Very good afternoon to everyone. Welcome in the class. And as we say, let's crack CSIR UGC NET together. You're watching this video under the platform of Unacademy. Unacademy is one of the India's largest platform for the preparation of CSIR UGC NET Examination. I'm your educator Farida Johar Bandukwala. I have a teaching experience of 10 years. I've qualified MP SLET examination in year 2018. If you're opting for Unacademy, what all things you are going to get? Let me just check whether I'm live or not. Let me just get the signal. Yeah. If you're opting for Unacademy, what all things you are going to get? You are going to get daily life classes. So with the help of daily life classes, you will be always in that momentum of studies. And in live classes, you can ask your doubts and queries with the faculty member. The whole syllabus is quite structured because you have to complete the whole thing in a limited span of time. So whole thing is quite structured. Then live tests and quizzes are taken so that you can evaluate yourself, and then unlimited access of all the videos whichever category you have opted for. Then with the help of Unacademy, you can attend the live classes. These live classes can be attended on YouTube platform. This is presently a YouTube platform where you are watching this video. Another platform is a special platform. So how to get the access of these special classes? You just download the Unacademy learning app. When you will download Unacademy, Unacademy learning app from the Google Play Store, you will have to make your account, and then you will be asked to fill the referral code. So fill my referral code FB01 and get an additional discount of attending the free live classes. Then Unacademy has another place where it conducts the classes, that is the Plus platform, and this Plus platform is a paid platform. And for paid platform, you need to pay a certain price. The amount is already quite pocket friendly. And if you're using my referral code, you will be getting an additional 10% discount. So do not forget to use my referral code FB01, and it is going to cost you 10,395 and 15,120 rupees. So this was a little introduction about Unacademy and myself. So the topic of today is vectors and plasmids. So first of all, we are going to see what are vectors, and if plasmids are used as vectors, what all things are required, and what all plasmids are used as in vectors. So first of all, we need to know that what is a vector, right? The first thing is vector. So vector is nothing but a DNA molecule. It's a DNA molecule. The DNA molecule. Now, if we are saying DNA molecules, so where are these vectors actually used? When we are coming across the word vector in such a college level class, when we have already studied science in uh, 10th, 11th, and 12th, vector relates to something related to physics. But here it's a DNA molecule, and this DNA molecule will be used in genetic engineering for production of recombinant DNA. So vector is a DNA molecule, and this vector is a DNA molecule which exists naturally, and its function is to replicate automatically. Replicate automatically or autonomously in an appropriate host cell. That is the function of a vector, to replicate. Like if there is a bacteria, and bacteria is governed by a genetic material, so this is its main genetic material, and this which gives it a specific quality of production of some chemical, or or a quality of production of pili on itself, fertilization. So that is a plasmid. It's a extra chromosomal material. So from where the vector thing is derived. So vector must replicate autonomously. It has the quality of replicating autonomously in a appropriate host cell. In a appropriate host cell, and into which the DNA fragments can be cloned and integrated for cloning. So the gene of interest gene of interest can be easily integrated into the vector for replication. Right? So a vector, when it is capable of replicating on its own, what all qualities it must have? What all qualities it must have? What all things are required for replicating autonomously? We are going to see that. So a vector must have an origin of replication. Then this origin of replication is denoted by ori that function in any of the host cell, and this vector is an extra chromosomal small genome, just like plasmid, phage or virus, and it can be used as a vector. So any extra chromosomal material such as plasmids, viruses, phages can be used as a vector. Now let us talk about the properties of a vector. Properties of a vector. Just give me a second. Sorry for the disturbance. So we are talking about the properties of a vector. So it should be able to replicate autonomously. Should replicate autonomously. Then a vector should be ideally less than 10 kb size. Ideally less than 10 kb size. Then a vector should be easy to isolate and purify. A vector should be easy to isolate and purify. Otherwise, in this step only, we will have to spend energy. So a vector should be easy to isolate and purify. Then it should be easily introduced into the host cell. Easy. So easy to introduce in host cell. Easy to introduce in host cell. A vector should have a suitable marker gene. A vector should have suitable marker gene so that it can be easily um, traced, that whether it has been incorporated or not, right? Then a vector should contain unique sites for the restriction enzyme so that in those places the gene of interest can be introduced. Should contain sites for restriction enzymes. So these all are some of the features or properties of a vector. Now vectors, they themselves can be divided into cloning vectors and expression vectors. So cloning vectors and expression vectors, they are used for three different purposes. That is, for cloning, they just need to increase their copy number and do not show any kind of expression. And for expression vector, what is their purpose? Their purpose is to produce certain kind of protein. So those are the expression vectors. Now, E. coli can also be used as vectors, and E. coli vectors are there. Then there are many things which are linked with that. So first of all, we'll be talking about plasmid as a vector. We have seen what all properties a vector should have. Then the topic is vector and plasmid as a vector. So plasmid. So a plasmid is basically a DNA molecule other than the bacterial chromosome, and it is capable of independent replication and transmission. So what is a plasmid? Plasmid is a DNA molecule. Plasmid is a DNA molecule, and this DNA molecule other than the bacterial DNA molecule, other than the chromosome, other than chromosome, other than chromosome, and it should be capable of independent replication. Capable of independent replication, right? So plasmids are DNA molecules which are found in the bacteria, and they are capable of independent replication. We haven't written found in bacteria, so they are found in bacteria, found in bacteria. Then they are capable of independent replication and transmission as well, and independent replication and transmission. So the plasmids are basically circular, are circular. They are circular in its structure, and they exist independent or become integrated into the bacterial chromosome. When they become integrated, they are called as episomes. Can exist independently or can get integrated into bacterial chromosomes. So when it gets integrated, the word it gets is episome. The word that it gets is episomes. Then plasmids, they vary in size. Their sizes are quite different for different types of plasmids. So their size may vary in size. That is 1 kb to 250 kb. Then an E. coli cell may contain up to 7 different kinds of plasmids. An E. coli can contain at a time up to 7 different kinds of plasmid. 7 different kinds of plasmids. Then plasmids, they are classified into several incompatibility group. Like if they are belonging to different compatibility group, they cannot exist in the same bacteria. So two or more different plasmids belonging to the same incompatibility group cannot reside into the same cell. Only one of them can be maintained in the cell. So plasmids can be categorized into compatibility group. Plasmids can be categorized into incompatibility groups. Into incompatibility. So if they are into the incompatibility group, they cannot exist together. So it is believed that it is believed that plasmids belonging to the same compatibility group share the same mechanism of replication control. Now, generally, the plasmids are not essential for the host cell except under specific conditions. So if they are living a harmonious life, they won't need any kind of plasmid. But if there is any kind of stress, they would need plasmids. So are not needed, plasmids are not needed by host cells in normal conditions. In normal conditions. Now the plasmids, they are not needed for normal condition. Okay, that is one thing. Another thing is they are of various types. So we are going to study the three main type of plasmids. Three types of plasmids. They are F plasmids, then they are R plasmids, then they are Col plasmids. F, R and Col. So F plasmid, F stands for fertility plasmid or conjugation plasmid. So F stands for fertility and it is responsible for conjugation. Responsible for conjugation. R plasmid, they carry gene for the resistance from antibiotics. So the fungus which releases the antibiotics for killing the bacteria. So bacteria has got its own defense mechanism, that is it has got R plasmid. So R is for resistance and R plasmid carry genes for resistance to antibiotics. Then Col plasmids, Col stands for colchicines. So they are responsible for killing the sensitive E. coli cells. So they also provide immunity to the particular kind of colicins. So Col plasmids, they are responsible for killing other bacteria. So colicin protein kills the sensitive E. coli. Kills sensitive E. coli. So these are some of the plasmids. So F plasmid is fertility plasmid and carry the transfer genes that promote conjugation. Example, there is a F plasmid of E. coli, which has got the F plasmid and another bacteria doesn't contain any kind of F plasmid. So it will transfer the F plasmid to the non-F plasmid containing bacteria and now both will become F positive bacteria. Then our plasmids are very much important in human health since they make bacterial cell resistant to specific antibiotics. So our plasmids are very important for human health. Then some of them have provided the antibiotic resistant genes used in the gene cloning. So an example of our plasmid is RP4 of Pseudomonas and species called even, and they have shown the function of resistance. Then there is one more type of plasmid that is called as degradative plasmid, and they are responsible for degradation of various chemicals such as toluene, then salicylic acid. So okay, let us write one more type of plasmid over here, that is degradative plasmids. Degradative plasmids. So they are responsible for metabolize unusual molecules, unusual molecules like toluene, salicylic acid, etc. Then there is one more type of plasmid, that is virulence plasmid. Though there are many types of plasmids, though we were just studying the three types in detail and two types in very brief. So virulence plasmid, they confer pathogenicity to their host bacteria. Confer pathogenicity to host bacteria. So do you remember that we have done TI plasmid and RI plasmid and agrobacterium mediated gene transfer? So virulence plasmids, their example is TI plasmid and RI plasmid. These are some of the examples of plasmids or the types of plasmids. Okay, so there are actually many different types, but we have seen these types. Now the plasmids are conjugative or transmissible. So they mediate the DNA transfer through conjugation. As a result, they spread rapidly among the bacteria of a particular population. So see, this is a bacteria having a specific type of plasmid, and these bacteria, they do not contain that specific type of plasmid. So they'll be existing like that in nature. So this plasmid can be transmitted very easily from one bacteria to another bacteria. So this type of plasmid which is easily transmitted from one bacteria to another, they are called as conjugative or transmissible plasmid. Conjugative or transmissible, conjugative or transmissible plasmids. Okay, then other plasmids are non-conjugative plasmids or non-transmissible plasmids, non-conjugative plasmids. So the non-conjugative plasmids, they do not mediate the DNA transfer through conjugation, and example of such plasmids are R plasmids, col plasmids. But some of the non-conjugative plasmids may also be transferred along with the conjugative plasmids when both of them are residing in the same cells. So imagine if a bacteria containing F plasmid also contain the col plasmid. So there are high chances of transmission of that col plasmid along with the F plasmid. So in conjugation or in existing together, there is a chance of getting transmitted altogether along with that is in the association. So non-conjugative plasmids, they can also be transferred. So when both of them, they reside in the same cells, the phenomena is called as mobilization, and it is dependent on the sequence of non-conjugative plasmid, that is the Nick bomb site of the non-transmissible plasmid. So we would write that transmission of transmission of non-conjugative plasmid along with conjugative plasmid is called as mobilization. It's called as mobilization, and this mobilization depends on Nick bomb site site of non-conjugative plasmid non-conjugative plasmid, right? Now there is another thing that is related with the plasmid, that is its stringent and relaxed replication. So stringent and relaxed conjugation. Stringent and relaxed conjugation. So each plasmid is maintained in the bacterial cell at a characteristic copy number, and mainly due to its replication control system. So in this respect, the plasmid are of basically two types, which exist in single copy and which exist in multiple copy. So astringent and relaxed conjugation is a term which is used for the number of copies of the plasmid. Um, criteria of existing as in single copy or multiple copies. Single copy or multiple copies. That is the stringent and relaxed conjugation. So the replication control of single copy plasmid is the same as that of the bacterial host cell. So they replicate and segregate with the bacterial chromosome, and this type of replication is called as stringent replication control. So single copy replication is along with bacteria's main replication system, and called as stringent conjugation. Other than that is multi-copy. So multi-copy replication is relaxed. The replication can occur even when the bacteria is not replicating. So it is different from the bacterial host genome. Replication is independent of bacterial replication. And called as relaxed conjugation. Not conjugation, it's relaxed replication. Relaxed replication. Stringent replication. Relaxed replication. So these are the things. Now we'll be talking as plasmid as a vector. We have seen what are vectors, then we have seen that what are plasmids. Now plasmid as vectors. Okay. So the next is plasmids as vectors. So many different E. coli plasmids, they are used as vectors. The natural plasmids, they have been modified shortened, reconstructed, recombined both in vitro and in vivo to create plasmid of enhanced utility and also with the specific functions. So the best one includes the gene which allows the selection of the recombinant DNA only, and some of the E. coli plasmid vectors are we are going to study. So these are the simplest cloning vectors and most widely used for gene cloning. Plasmid vectors, we should write one sentence with this. They are the simplest. They are simplest. They are simplest cloning vectors. They are simplest cloning vectors and are most widely and are most widely used for gene cloning. So they are obtained from plasmids and they are not used directly. So natural plasmids are modified. Natural plasmids are modified. Then they are shortened. Then they are reconstructed. Then they are recombined and recombined for production of plasmid as a vector or the one which can be used, right? So this is the thing. After that, how to select the recombinant DNA, that is the thing we are going to see, and then we'll be talking about uh, the examples of the vectors. So okay, now there is a question by Shruti Garg and she is saying, uh, where is that? She is asking that um, which is the smallest sized vectors. So there are many newly obtained vectors which are of small sizes and which are used. So you cannot actually go for the one which is the smallest vector. We look for vectors in terms of how many kb of DNA it can actually incorporate inside itself. So you are saying PBR 322, so it is not the smallest vector actually. Whenever you will come across the question which is asking you which is the smallest vector amongst this, so you can go for it. But if you'll go for looking for the smallest vector available in today's time, so you cannot, ma'am, I am not asking. Okay, Shruti Garg is saying, okay, okay, fine, you cleared it. Otherwise, I would have elaborated that. That now selection of recombinant DNA, recombinant selection of recombinant DNA. Now, recombinant DNA, whether the vector has actually taken the gene of interest or not, so those all things, selection of recombinant DNA is one of the most important thing in this whole story. So it may be pointed out that when an experiment is performed and a DNA insert is inserted into the vector, they the following two types of vector molecules are actually obtained. The vector molecule which is going to contain the DNA insert, and the another one will contain just the vector sequence. So now how to differentiate those, that is the criteria in selection of recombinant DNA. So we'll write that after experiment two different vector molecules are obtained. Two different vector two different vector molecules are, oh okay Shruti, I have read your comment. After experiment, two different vector molecules are obtained. So these are, first one is the vector which is going to contain the DNA insert, vector with gene of interest. So the DNA insert, I'm calling it as gene of insert or recombinant DNA, vector with gene of interest or rDNA. You can call it this way. And the second type is, most others will contain only vector sequence, and we are going to call it unaltered vector. Now how to differentiate that which vector has got which one is the one containing the rDNA and which one is the unaltered vector. So for that, the selection of recombinant DNA is done. So the mixture of vector and recombinant DNA molecule is used for transformation of the host cell. So some host cells are going to receive the recombinant DNA and the others one are going to get the unaltered vector, right? That is how it is going to happen. So in cloning experiments, it is critically important to have a marker sequence which is actually going to give us the indication that yes, this vector is actually transformed and this vector is not transformed. So selection of host cells transformed by recombinant DNA is easily achieved by placing two selectable markers. Remember this: one for antibiotic resistance genes such as ampicillin or tetracycline, any of the. So the DNA insert is integrated within one of the two selectable markers. In the first category and in the another category, there would be no insert. So both of the things would be intact. So this can be achieved by, this can be achieved by, this can be achieved by using two selectable, two, two selectable markers. So gene of interest introduced would be introduced into the one of the selectable marker, and this is how the whole story of selection would go on. So now the unaltered vector would contain both of the resistance gene, and the vector with gene of interest is going to contain just one intact antibiotic resistance gene. So it won't be surviving on a media containing both of the antibiotics, and the host cell containing the unaltered vector is going to survive on any media because it has got the resistance gene. So that is how we'll do the comparison and we will identify using transformation process. Yes, Shrutika. So that is how we are going to select the different types of transformed cells, that which one has been transformed and which one has not transformed. So we'll be talking about some of the examples of plasmid as a vector, and this we are going to do in a very quick way, because these things you should not emphasize on unnecessary things. You should just specifically remember 4 to 5 points which can be enough for the question. So first one is PSC 101. So this is the earliest plasmid vector which contains the replication module for replication of E. coli. So tetracycline resistance gene for resistance to the tetracycline, and single recognition site for the restriction endonuclease of E. coli was there in PSC 101. So what we can do is, I'll provide you with a table. In my next video or in the same video, I'll share the link of the tabular form of the plasmids which are used as a vector. So that thing we can do. So you just see over here some of the names of plasmids which are used. So I'll make a table and I'll just try to put the important things in that. So other than that is PBr 322, which is a ideal plasmid vector. So PBr 322 is widely used and it's an ideal plasmid vector. Why is it a plasmid of ideal choice? Because minimum amount of DNA less than 10 kb is there to avoid the problems such as DNA breakdown or purification. So its size is small, that is less than 10 kb, then it undergoes relaxed replication. It has two selectable markers and it has one recognition site for at least one of the restriction endonuclease. So PBR 322. After that, PBR 322 is studied widely at various universities. They have this PBR 322 specifically written in their syllabus because it is widely used. Then there is this PUC 18 and PUC 19. So PUC series of vectors are actually derivatives of PBR 322 only, and they are much smaller. You would be shocked to see that they are even of the size 2.7 kb. So Shruti, dear, you were saying that PBR 320 is the smallest one. So I told you that do not look for the smallest one directly, because then you will come across a very new name, because vectors are getting invented or developed each and every day. So the one which is given in the category of the question, you have to select from that only. We cannot exactly go for a smallest one. So PUC is a vector plasmid vector, which is actually derivative of PBR 322 only, and it is much smaller than PBR 322, and you can see the size over here. Then the another one that you need to know is PGEM TZ. So this plasmid vector is very similar to PUC only, and exactly of same size. Just the difference where it is different from PUC is in the resistance markers and those all things. So like this, I've explained you the whole thing and just the important points. So what all the list which I'll be giving or table which I'll be giving. So in that we'll be writing the name of the plasmid vector, we'll be writing its size, we'll be writing that how much amount of DNA it can carry, which are the resistance markers, and which what is its origin site, and some 2-3 important points if it is linked with it. So I'll just make a table and I'll share over here. So do not forget to come back to this video for getting that table. Okay, so I think like this, these are the plasmid vectors. So in today's class, I've just included the plasmid vectors. We'll be doing yeast artificial chromosomes. We'll be doing cosmids. Be doing phagemids. All those things also we'll be doing. But do not forget to remember the basics, because the questions they ask is usually from the basics. So astringent and relaxed replication, conjugative or transmissible plasmids, those all things are quite important according to the exam, because if you're going by the name, then all the time new plasmids are getting invented. So we'll be just talking about the famous plasmids, famous for the examiner, which examiner might ask you in the question examination. So that is the way we are doing, and this is it for now. And I hope you people have seen the classes of modification and modification enzymes needed for rDNA technology. I've described 7 enzymes which are needed for modification in genetic engineering. Try to see those classes. And Shruti is asking me, ma'am, please tell me about your class timing and topics. See, tomorrow morning 10:30, every day 10:30 I go live in the morning, talking about the CSIR crash course, and evening 5 o'clock I go live. And as far as you are asking me that what topics I am going to teach you, so let me just tell you that what topics I'll be teaching you in the coming days. Let me just open the sheet and I'll be sharing with you. Just give me a second. So the topics which I am going to teach you further in the morning class would be ELISA, flow cytometer, then RIA, then isolation and purification of RNA and DNA, 2D gel electrophoresis, isoelectric gel focusing, protein sequencing, and much more. And in the evening class, I'll be teaching you in tomorrow's class at 5 o'clock. I'll be talking about yeast artificial chromosomes, then cloning and expression vectors we'll be talking in the class of day after tomorrow. Then we'll be talking about DNA labeling, DNA sequencing, DNA fingerprinting, southern blotting, northern blotting, and western blotting, fish, fluorescent in situ hybridization, then RAPD, gene therapy, RFLP. Those all things we are going to discuss in further classes. So there is a whole batch which I have whole list of topics which I'm thinking that can be important for you. Those I have prepared, and we'll be talking about them. And I hope to see you uh, tomorrow morning and in the evening as well. So Shruti Kark, any Telegram channel so that I can relate and prepare accordingly. Yeah, Shruti, I hope that you have heard that what all topics I'm going to teach you in the further classes. So talking about my Telegram channel, yes, I have a Telegram channel. You can join over there as well. So you can join over there by you can look for the name F Johar. I'll be asking for sharing the link of my Telegram channel as well over here. So do not worry for that. And whenever you are back in the class, look for the uh, table which I'll be sharing. Okay, I hope the things are quite clear now. And as we say in the end, let's crack CSIR UGC NET together and GATE 2021 together. And if you haven't subscribed our channel till now, hit the bell icon for getting the notification and use my referral code FB01 for getting a 10% discount in the Plus subscription, and go and attend the special classes as well. For attending the special classes also you will need to fill the referral code of an educator. So fill my referral code there, that is FB01. And if you really like my video, please give it a thumbs up. And lastly, it's a thank you, and hope to see you tomorrow 10:30 for CSIR crash course and 5 p.m. for the GATE crash course. So till then, thank you very much and keep on studying and be happy. So finally, it's a bye from